this type of epigenetic regulators, so mainly small RNAs. So I'll move on. First, just a little bit of background. So in the Australian wheat belt, increasing temperatures and decreasing rainfall have been big challenges for crop production. So usually the water deficit stress would occur around springtime, and that is considered as pre-anthesis because it usually happens before flowering. And then we could have a heat stress problem after flowering, considered as a post-anthesis heat stress. So water deficit stress and heat stress, they often occur at the same time in mid to late growing season. So these combined stress episodes often cause significant yield penalty and growing quality downgrades. So one of the opportunities that we can use for crop improvement is in molecular breeding, is this type of epigenetic regulators. So we'll call Microarrays. They can confer superior grain quality and high yield stability, even under abiotic stresses. So we can aim to utilize such genetic and epigenetic diversity in the current germplasm to improve stress tolerance. So a bit more details about plant microarrays. So they are a type of small non-coding RNAs. They can regulate gene expression, so the expression of their target genes on the post-transcriptional level, mainly through triggering messenger RNA transcript cleavage and or translational inhibition. So microarrays have been a new target for improving stress tolerance in crops in the last few years, because they're involved in many developmental and signaling pathways, and microarrays have expression patterns that are specific to stress type, tissue type, genotype, and developmental stage as well. Because of the gene silencing effects of microarrays, when a microarray expression is induced under stress, that can lead to the repression of their target gene, which could be negative regulators of stress adaptation. And when a microarray expression is reduced under stress, and that could allow for the accumulation of their target gene expression, which often could be positive regulators of plant growth or stress adaptation. So we proposed the concept of smarter breeding in this review paper we did for Transient Plant Science a few years ago. So here we basically highlighted all these key microarray and messenger RNA modules that you can use in breeding or molecular techniques to manipulate certain traits that are of interest, for example, flowering time or tiller number or even green size and quality. But in cereal crops, our current knowledge about microarrays mainly come from species like rice, barley, and bread wheat. We actually know very little about the microarray regulatory networks in tetraploid wheat, mainly durum wheat. So that is the focus of my project. So for this project, the research framework mainly has three components. So the first, where we looked at the genotypic response of Australian durum wheat to water deficit stress and heat stress. So here in the Glasshouse, we looked at the crop performance across a range of Australian durum varieties and breeding lines. And we studied their physiological traits, morphological traits, yield and growing quality traits under different stresses. So in the second part, which is what we've been working on mainly in the last year or so, is where we used next generation sequencing technique in a few different ways to analyze the stress regulatory networks. So here we use small array sequencing, degradome sequencing, and transcriptome sequencing. And that enables us to identify all the regulators of stress response and grain development. And the second part will be the main part of my talk today. So in the last component, we aim to look at specific, like key microarray and messenger RNA modules that could contribute to stress tolerance and grain quality, where we'll be looking at their molecular activities and their expression profiles, and to basically associate their molecular functions with crop performance that we can observe on the physiological or yield level. So first, just a little bit about the first component. In the Glasshouse, we use 10 Australian durum wheat genotypes. So that's 5 commercial varieties and 5 University of Adelaide advanced breeding lines. So we had 4 treatment groups. So control group, water deficit stress group, heat stress group. So these 2 groups are just a single stress. And then we had the last treatment group is a combined stress group where the plants were treated with water deficit stress plus heat stress. So in terms of the stress application, the control group was well watered from germination to maturity and under standard growing temperature. So water deficit stress group had the pre-anthesis stress treatment where the water deficit stress started at a brooding stage. So what we did is that we withheld watering, so keeping the soil water content in the pots at half of the field capacity all the way from brooding to maturity, and that's under standard temperature as well. So for the heat stress group, the plants were well watered from germination to maturity, and the heat stress was applied after flowering at 5 different time points every 10 days. So that's 5, 15, 25, 35, and 45 days post-anthesis. So at these 5 time points, the plants were moved to a heat stress chamber where the temperature was 37-27, and the plants stay there for 24 hours, and then they go back to the standard growing condition between these stress treatment times. So for the combined stress treatment group, so the plants had the water deficit stress started at brooding as well. So from brooding to maturity, the field capacity was kept at 50%. And then the heat stress was applied at these 5 time points as well for 24 hours by moving them to the heat stress chamber. All right. So during the whole life cycle, we took a few measurements. So for the physiological traits, we looked at chlorophyll content, stomata conductance, liver relative water content at these 5 time points. And for the morphological traits, we looked at plant height, main spike lengths, flag leaf width and length, and then we'll measure the standard growing, yield components such as grain weight, grain number, etc. at harvest. For grain quality, we mainly look at 3 traits, so protein content, starch content, and the phenolic content. So for durum wheat, to achieve the best price, you want to have a minimum protein content of 13% for the growers. We also took photos along the way at different stress time points just to compare how they look between different stress groups and between different genotypes. Also took photos of the grains as well when we sampled them. So yeah, for samples, I sampled the flag leaf and the developing grains on the main stem. All right, so here what I have is the photos I took along the way. So on the left, this panel is a stress-sensitive genotype, line 6 is just a breeding line. And this breeding line was the most sensitive to the water deficit stress and heat stress. And on the right, what I have here is DB Aurora. DB Aurora was ranked on top in terms of stress tolerance, so it was most tolerant to the stress. And then for the photos, you can see I took photos at 5 different time points from day 5 to day 45 after flowering. And if you look across this way, that's the control group, water deficit stress group, heat stress group, and the combined stress. So if you compare across between the 2 genotypes, it's very easy to see that the difference between DB Aurora and line 6, you can see Aurora had a lot more tillers, and especially under the combined stress group, and it had more green leaves as well, where in line 6, there was a lot of yellowing in the leaves, which reflected on the chlorophyll content, the measurement was a lot lower. So if you are interested to look at any of the results, they are published in scientific reports last year. So I'll move on to the molecular component of my project, where we use different sequencing platforms. So the sample sets used for next generation sequencing are from the 2 genotypes that we just looked at. So a tolerant genotype and a sensitive genotype. So the tissues we used were flag leaf and the developing grains from the main tiller. So the developing grains were peeled out of the tiller, so we use the grains rather than the whole head. So yeah, we included the 4 treatment groups, and the development stage was these 5 time points. And you can see the photos I put here is what the grains look like at these time points. So at day 45, they're pretty much close to maturity, if not already. Right, yeah. So we use 3 different sequencing platforms. I'll explain each in a little bit. So small array sequencing, we use this to discover conserved and novel microarrays in durum wheat. So conserved microarrays are the ones that you can find almost in most of the plant species. And the novel microarrays are the ones that haven't been identified before and could be durum specific. So we were the first to use the new durum genome assembly, which was released last year for microarray discovery. This is the first complete genome that we have, and which really improves the bioinformatics results. So from this, we can identify differentially expressed microarrays that are responsive to stress or have genotype dependent pattern or time point dependent pattern. So for transcriptome sequencing, that's just standard gene expression profiling at the transcriptome level. Here we can find differentially expressed genes responsive to different stresses, and the ones that with genotype dependent pattern between the tolerant and the sensitive genotypes. We also use geoanalysis and KEC pathway analysis to functionally annotate the genes that we found that have a significant differential expression pattern. And for the last part, which is the Decoding sequencing, probably not all of you are familiar with it. So it is based on the parallel analysis of RNAmes. So if you remember, I explained before, microarray reduce the expression of the target genes. So if you look at here, when a microarray finds its target, it binds to it, and that could lead to messenger RNA transcript cleavage. And it basically will be broken to 2 bits. And with Decoding sequencing, it can globally capture and sequence the uncapped 5 ends of the cleaved messenger fragments. So you will be able to validate the cleavage sites. And currently, it's the only high throughput method that you can do that. And our group was the first, I think, still the only group that have used Decoding sequencing in durum wheat before. A lot of people have used this technique in rice and bread wheat and barley, but not in durum wheat. Okay, so I'll get into the results. First, let's look at the small array transcriptome. So with the small array sequencing, we made 240 small array libraries, which is a lot. Because usually, if you look into the papers, the normal numbers, you probably see people do 12 libraries or 24 libraries. So with the 240 libraries, we captured 2 different genotypes, 4 treatment groups, and that's 2 tissue types and 5 development time points from day 5 to day 45. And we used 3 biological replicates. So that generated over 2.4 billion reads. Took me almost a full day just to download the sequencing data. So for the bioinformatics pipeline, we use CLC Genomics Workbench and this ACGT MIR program. That's a commercial program developed by people in China. So if we look at what I've shown here is the length distribution of the sequencing results. So small array are very small in size, so usually between 18 to 24 nucleotides. And usually you see classic 2 peaks at the 21 nucleotide length and the 24 NT length. So in the flag leaf libraries, because we use 2 tissue types, in the flag leaf we found over 500 microarrays, and 55 of them are novel. So they've never been discovered before, and they could be specific to durum wheat. In the developing grains, we found slightly more microarrays and also 84 of the novel microarrays. So next, we will look at the expression specificity of microarrays in the leaves. So I'll take a little bit of time to explain this figure. So what we have here is different Venn diagrams of durum microarrays identified between different biological groups in the flag leaf. So you see this type of figures a lot for next generation sequencing studies. So the panel A here is what we found in the stress-tolerant genotype DB Aurora, and panel B is about the stress-sensitive genotype line 6. So if we just look at one little Venn diagram here, so the TL here stands for leaf libraries from the tolerant genotype DB Aurora, and the numbers here 5, 15, 25, 35, 45. That's the days post-synthesis. That's the time point where we sampled. And if you look at the letters here, CG, HS, WH, and water, WS, that's the 4 treatment groups as I typed in here. So when you look at the numbers, if you look at the ones in the middle, so this 186, that means 186 microarrays, you can find the expression across these 4 biological groups. While if you look at these numbers here, there were 12 microarrays that were only expressed under the control condition, and 5 microarrays were only expressed under the combined stress treatment. The key message here is that if we just look at the numbers in the middle, DB Aurora actually has less commonly expressed microarrays across treatment groups when compared with line 6. So you compare these numbers, you see the numbers is always lower in DB Aurora, except for day 45. So these are the results that came out of the flag leaf. And we did the same for the grains. It's the same, you can compare across treatment groups and look at different time points. And you actually find the opposite trend in the grains, where the number here is actually higher in DB Aurora in the stress-tolerant genotype. So this tells us that you actually see different patterns between the 2 tissues. And in the stress-tolerant genotype, the leaf microarray population had higher expression specificity. That's why you have a lower number that are commonly expressed. But in the grains, the microarray population actually had lower expression specificity. And that could be just related to the different functions that the microarrays have in different tissues. So move on to the next, where we looked at the stress-responsive patterns of microarrays. So the microarray expression level were compared across the 4 treatment groups based on the normalized read count in each library. So these are there's going to be a lot of numbers in my talk. Please just bear with me. If you want to dive deeper into the data, the results are published in IGMS just last month. Today, I'm just giving you a quick Snapchat, so bear with me. So on the left, what we have here are the numbers of stress-responsive microarrays that we found in the leaf. And in DB Aurora and in line 6, you find different numbers of stress-responsive microarrays at different time points. And here is what we found, the number of stress-responsive microarrays in the grain tissue. And again, between the stress-tolerant and the stress-sensitive genotype, you find different numbers of stress-responsive microarrays as well. What's interesting here to note is that in the leaf, you actually find that you have more microarrays that are stress-responsive at the beginning of reproduction and at the end when the grains are almost getting to maturity. But in the grains, you find the higher number of stress-responsive microarrays at earlier stage, 5 dpa, 15 dpa, and 25 dpa. All right, so the next, we looked at the genotype-dependent expression pattern of microarrays in the flag leaf and in the grains. So what I have here is, so on the y-axis is the number of the microarrays with the genotype-dependent expression pattern. So basically what we compared is, if you look at here, we compared the microarray population under the control condition at day 5 between the stress-tolerant genotype and the stress-sensitive genotype. And you have 120, what we call upregulated microarrays, sorry, over 120 downregulated microarrays, and you have slightly over 40 upregulated microarrays. So the upregulated microarrays are the ones that had higher expression in DB Aurora, and the downregulated ones are the ones that had lower expression in DB Aurora. So a very obvious pattern here is that you can see the red colored bar is always higher than the blue colored bar. Doesn't matter if it's under the control condition or water deficit stress condition, you always have more microarrays, a higher number of microarrays that had low expression in DB aurora. So they're more downregulated than upregulated. So this is what we found in the flag leaf. But if you look at what happens in the developing grains, is the opposite trend, except for you do see a few exceptions here. But the majority is that in the grains, there are higher numbers of microarrays that had higher expression in DB aurora. So they were more abundant in DB aurora. And so the trend is that you see different patterns between the two tissues again. And that could, again, just because of the biological functions that different microarrays play in the flag leaf and in the developing grains. All right, so I'll move on to the transcriptome sequencing results. So we sequenced 16 transcriptome libraries, and then we only did that on the day 5 after thesis samples. So we generated over 1.3 billion reads, and we mapped them to the reference genome. So in total, we had nearly 50,000 genes identified in the leaf and over 50,000 genes identified in the grains. So what I have here is box plots. So basically, it shows the distribution of genes on the left here and the distribution of transcripts among the 8 flag leaf libraries with a 5-number summary as shown in the box plots here. So for transcriptome sequencing, the standard practice that you will normalize the gene expression with this term we call it FPKM, so fragments per kilobase million, and you use that as a normalized count when you compare the expression across your different treatment groups or different genotypes. So as you can see here in the box plots, the distribution is relatively stable across the different libraries. And that just shows that you didn't have anomalies in terms of the library prep. All right, so move on where we looked at the stress-responsive genes. So we analyzed the gene expression level under each type of stress compared with the control group. So I'm going to have a lot of numbers here again. So in the flag leaf, in the 2 different genotypes, so tolerant and sensitive, these are the number of genes that we found are responsive to different types of stress. So single water stress, heat stress, and combined stress. And the numbers are different when you look at the developing brain tissue. So one trend that we found here, if you haven't noticed already, is that you always have the highest number of stress-responsive genes under a single heat stress, and you find the lowest number under water deficit stress. So so the trend is that heat stress actually triggered the highest number of stress-responsive genes, and the lowest number of stress-responsive genes were found under single water deficit stress. And the reason is that the heat stress we applied was a rather intensive stress shock. So, and it was rather transient in nature because we just moved to a heat stress chamber for 24 hours and then it comes out. So it's almost like a heat shock. But for the water stress that we applied was a mild water limiting stress, and it started at booting, and then it lasted all the way to maturity. So the plants would have a little bit of time to adapt to it along the way. So what we did next, after finding all of these stress-responsive genes, we want to look at, okay, what do they actually do? What are they? So a lot of them are transcription factors. Some of them are plant growth regulators. A lot of them are related to hormone signaling pathways. And many of them are key metabolic enzymes, especially for the ones that we found in the developing grains. So next, what we did is we performed geo-enrichment analysis and KEC pathway analysis of these stress-responsive genes. So basically, it gives us an overall summary and visualization where you put all of these genes into different categories and then see what are the major functions. So for results, I have an example figure here, which I'll explain in detail. So these are what we call scatter plots. So here what we have is the scatter plot showing the geo-enrichment results of stress-responsive genes under combined stress in the flag leaf. So this is just an example. What we have on the left is the enrichment results in dB aurora, and what we have on the right is line 6. So if we look at the y-axis here, so these are the top enriched geoterms that had a significant value, and the plots, they use the scatter plots you see here. The size of the plot, how big they are, indicates the gene number, stress-responsive genes that are enriched under a specific geoterm. And what you see here on this way, where we have rich factor, the rich factor represents the degree of enrichment is calculated by the ratio of stress-responsive genes that have this geoterm and the total number of genes that have this geoterm. So basically, the higher the rich factor is, you have a higher degree of enrichment or representation. So by doing this, you can see how these genes, stress-responsive genes, are distributed in terms of their geofunctions. And you can compare that between two genotypes. You can tell that some of the geoterms are shared. You can find both of these terms in dB aurora and line 6. But some of the terms were genotype specific. For example, responsive to hydrogen peroxide was only found in dB aurora, and RNA processing was only enriched in line 6. So, so as I said before, this is just an example when you look at, all right, I just want to see what happens under combined water stress, combined water deficit stress, combined water stress and heat stress in the flag leaf. What you can actually do is that you have all the stress conditions put together. So that's water deficit stress, heat stress, and combined stress. And you have dB aurora along this way, and you have line 6 along this way. So on the left, these are the scatter plots for geoterms. And on the right, you have the scatter plots for the KEGG pathway analysis. So what you can do is that you can compare this way, and this tells you what geoterms were enriched that had genotype dependent pattern. And if you compare this way, you can tell what geoterms were specific under a certain type of stress. What's interesting is that we found that certain pathways were only activated under combined stress condition. They were not triggered under either of the single stress conditions, but they were activated under combined stress condition. This tells us that there were possible interactions between the two types of stress at the transcriptional level, which have been observed in other crop species as well. So I'll move on to the to the genes that with genotype dependent expression patterns. If you remember where we look at the blue bars and the red bars for the microarrays, you actually see a similar pattern here. So in the flag leaf, these are the number of genes that we found have genotype dependent pattern under different treatment groups. And in the flag leaf, the blue bar is always higher than the red bar. So you have more genes that have higher expression in dB aurora. But in the flag leaf, you see an opposite trend, except for under the control condition, where you have a higher number of genes that had low expression in dB aurora. So this tells you the genotypic pattern between the genotypic pattern that you can find in the two different tissues. And again, similar to the genotypic microarray, that could just be due to the different molecular functions that they have in different tissues. All right, so on the when we did the transcriptome sequencing, we also had a quick look at the AS events, alternative splicing. So AS is a process that basically enables the same messenger RNA to produce different proteins variants that may have different functions. So there are 12 major AS events in crop species. So here we looked at these 12 major AS events, as you can see here, and across different genotypes and the different stresses, which I put here in terms of their library name. So the obvious pattern is for these two types of AS events, which is TSS, alternative transcription start site, and alternative transcription termination site. So these two categories had the highest number of occurrence. And if you look at across the 4 treatment groups, where you have control, heat stress, combined stress, and water stress, So the blue ones are the stress-sensitive genotype and the red ones are the tolerant genotype. Regardless of genotype, you find that water deficit stress actually reduced the number of AS events. You always have the lower number under water stress, while heat stress actually triggered the highest number of AS events. So this is not really the focus of our project, but it's just a very interesting phenomenon to note because no one has ever discovered this type of pattern before. All right, so I'll move on to the next to mRNA degradome sequencing. So we did 16 libraries again. So that generated nearly 1 billion reads, and the reads were mapped to the reference genome. So basically for decorative sequencing, we're looking for the degradome signatures that are consistent with a microarray guided cleavage event within the mRNA transcripts. So in the flag leaf and in the developing grains, we found these are the number of microarray-induced mRNA degradation sites in the two different genotypes. So there are slightly less degradation happening in the grains comparing to the flag leaf, if you look at the numbers here. So what I have here is called the target T-plots. This is the just the standard way to visualize your degradome data is to look at the distribution of degraded mRNA transcript tags along the full length sequence. So let's just look at one figure here on the left. So here along this way from 0 to 1200, so this is the full length transcript of this gene, which is the DREB transcription factor. And this mRNA was targeted by microarray 398A, and the cleave position is at 435. So all of these peaks are the peaks of degradome reads, and then the red dots indicate where microarray directed the cleavage sites. So it's around the 435. So you can see that sometimes the microarray guided cleavage could be the top peak and sometimes it's not. And you basically rely on bioinformatics tool to verify whether it was a microarray guided cleavage site or not. All right. So the next, after we find all of these microarrays guided cleavage sites in the mRNA targets, we want to look at, okay, what do these mRNAs actually do? What are their biological functions? So a lot of the mRNA targets were major transcription factor families, as I listed here. A lot of them are metabolic enzymes, especially for the ones that we found in the developing grains. And there are a lot of genes that are related to hormone signaling, and a lot of them are major families of protective proteins such as SOD, POD, and a lot of them are heat shock proteins as well, especially for the ones we found under heat stress treatment. All right, so now we have all of these data. We have data from small-array sequencing, mRNA sequencing, and degradome sequencing. What do we do? How we combine them? So this probably is the key part of the analysis, that we want to combine these three types of data to integrate them together to find the key information, find the microarrays and mRNAs that are actually important. So based on the silencing effects of microarray regulation, basically what we're doing here is that we look for the pairs with a significant antagonistic expression pattern. So if a microarray expression goes down, the mRNA expression would go up, and that allows for the accumulation of positive regulators. And if a microarray expression goes up, we expect for the mRNA target expression to go down, and that could be the repression of some negative regulators. So there are three major steps to do this in terms of data analysis. So first, what we did is that we found all the microarray and mRNA pairs that can be validated by three types of sequencing data at the same time. So basically you can find reads counts in these three types of sequencing data. And after we find all of these pairs, you narrow down to the ones that actually had a significant stress-responsive expression pattern at p-value at 0.05. So that's for both the microarray and the mRNA. So they both have to have a significant expression pattern. And then from the significant pairs, we narrow it down again to find the ones where they actually have a negative correlation. So if one goes down, the other one goes up, and if one goes up, the other one goes down. So that's how we pinpoint the ones that have significant antagonistic expression patterns. And these are probably the ones with the most important biological functions. So here are a lot of numbers again. So these are the number of the ones that we managed to find after those 3 major steps. So in the flag leaf, these are the ones we found in DB Aurora and line 6. And in developing grains, these are the number of microarray and messenger pairs that we found under different stress conditions in the 2 genotypes. So the next step would be, all right, what do they do? What are the actual functions that could be specific to genotype, stress type, and tissue type? So all the results are published. You can find a detailed list of what they are. So the gene ID, their functions, the geoanalysis, and CAC pathway analysis for all of these genes, if you're interested to have a closer look. And then from here, what we did is that we want to draw a map, like a summary map, to find the ones that can contribute to either stress adaptation or grain quality during reproduction. So what I have here is a summary figure. So basically, we highlighted some of the key microarray and target gene pairs that could be important to different steps of stress response. And so you can see some of them are related to signal perception because of the target genes that microarrays targets, and some of them are related to signal signaling, where you have the ones, for example, oxygen response factors, or the ones that are related to, for example, the CBL, CIPK pathways. And of course, you have this major transcription factor networks as well, where you have lots of different microarray family members that target different transcription family members. So what's interesting to look at here, so the red color of the microarray, that means this microarray expression was downregulated in the stress-tolerant genotype. And the green color of the target gene means that this target gene was upregulated under stress in the stress-tolerant genotype DB Aurora. So all of these different modules could eventually contribute to different aspects of the cellular or physiological adaptations. For example, you have some genes here that are heat shock proteins or ROS scavengers, such as SOD and POD, and that could contribute to cellular redox homeostasis. And all of these different cellular changes or physiological changes would eventually contribute to water deficit and heat stress tolerance in the plant. So this is a whole summary of what happens in the stress adaptation process. We also did some of these maps, basically summarizing the ones that could contribute to grain quality analysis from flowering to maturity. So here what I have is cytoscape plot of the key microarray and messenger arrays that are involved in starch accumulation. For example, you have target genes that are related to starch synthesis. And what's important to note here is that some of genes are rather sit in the middle of this regulatory network because they are regulated by many different microarrays. And their microarrays, for example, look at here, it regulates different genes at the same time that are involved in starch accumulation. So this is the starch accumulation plot. We also did one for the protein accumulation. So this one looks a little bit cleaner. It's easier to find what are the ones that are important. For example, here, the microarray 167 could be really important because it's regulating 4 different genes at the same time that are related to protein production. And here we also did the ones for the carotenoid biosynthesis and phenylpropanoid pathway as well. So again, the results are published if you want to have a closer look. So what's interesting to note in this plot is that you can see that the regulation between microarray and their target genes, they are there's this multiple to multiple relationships. They are not one-on-one specific, and there are always interactions between different modules. And all of them contribute to fine-tuning the expression of the functional genes that actually make the useful proteins. So, so the key here is to focus on the microarray and the messenger candidates that connect through all of these different pathways. So these candidates will provide a good entry point into improving the production of nutrient partitioning in cereal crops, as well as the coordination of different biological pathways required for grain development and stress adaptation. So for ongoing work and future directions, what we're doing is that we're looking into validate some of the key modules that we found in those maps that contribute to stress tolerance or grain quality. And we're also going to look at them into a broader range of germplasm to determine there are actually possible significant associations between mRNA dynamics, gene expression, and crop performance on the physiological or the yield level. Another topic that we're interested in is that we're actually looking at how this sort of a regulation can be passed down across generations. Do small RNA regulated stress response actually have a transgenerational effects? So so this year we're in the third generation now. We actually do see some difference between the ones that have been stress primed in the previous generation and the ones that haven't been stress primed in the previous generation. All right, so at last, I would like to acknowledge the funding bodies of the research and my mentors, Dace and Amanda, and the group members in our group. Thank you. All right.
Thank you, Haipei. Could people please type any questions they have in the Q&A box at the bottom of your page? I do have one question to start us off.
Mm-hmm.
Why do you think under the combined stress there were, why under the combined stress were there less stress responsive genes than under the heat stress alone?
Yeah, I think because the way that we did the stress is that water stress actually occurred before flowering already, and then the heat stress hit after flowering. So rather than the single heat stress, those plants were basically just living a happy life without any stress and then suddenly put into a stress chamber. And that, because it's such an intense heat shock, that triggered a lot of response immediately. But for the combined stress, because the plants actually had time to probably like at least 20 days under this mild water stress before the heat stress hit, and because there are so many crosstalk between the water stress and heat stress, some of the genes are probably already tuned to the level where it would contribute to both water stress and heat stress adaptation. So we didn't need to change again, if that makes sense. So yeah, I guess it just comes back to the interaction between water stress and heat stress.
Okay, I've got two questions from Penny Tricker. The first one is, were any of your mRNA modules durum specific, or were they all conserved microRNAs? Yeah,
so that's a very good question. So for the ones that we showed in this sort of cytoscape plus here, these are all the conserved ones. You would see this kind of a 3-letter at the front, and this basically stands for the species where they were annotated in the plant microarray base. So we do have novel microarray and messenger RNA modules that are durum specific, that are novel, but we just didn't put them here in this summary, but you can find them in the supplementary tables of the paper that we published. Yeah, they're definitely novel ones as well.
Okay, the second one from Penny is, the number of genes identified in your library seems low. Do you have any idea why this is?
Yeah, we were wondering that as well. I guess we don't really have a previous study that we can compare to, because we are the first group that actually used the first durum wheat genome assembly, which was just published in 2019, and we're the first to actually use that as reference genome for transcriptome sequencing. So we were actually, what we use in terms of the parameters is just standard parameters. And I guess that could just due to maybe the genome was not actually that fully annotated as much as we expect. And this is just the number of genes that we can find. And like we would like to identify more genes, but this is just what we can find using that Saviva1 as a reference genome.
Okay, a question from Scott Bowden is, do you have any data to say that the identified microRNA mRNA modules are responsive to removal of the stress?
Oh, that's an interesting question. Yes, we thought about what we have is you probably wouldn't be able to find that for the groups where we had the water deficit stress, because that's just from booting to maturity, the stress was always there. But for the heat stress, is really interesting. You actually can look into the ones that could be related to stress removal, or the ones that could be related to reoccurring stress memory, because the stress was applied, as I said, day 5, and then the stress was gone. And then day 15, you had the stress again, and then 25, 35, 45. So you had this kind of 5 reoccurring stress. So if you investigate the expression between these time points, you might be able to find some information. But I guess the answer is the best, if you want to look at how that responsive to the removal of stress, is that if we had a sample, let's say at day 10, between 5 and 15, that might give you a better idea. Yeah, does that answer your question? I guess it will give you some sort of idea, but the setup was probably not ideal to look at the ones that are responsive to stress removal.
Okay, we've got a question from Rakesh David.
Yeah.
A large proportion of your small RNA sequencing is in the 24 NT range, which are known to be involved in transcriptionally silencing transposable elements.
Yes.
Do you know what proportion of differentially expressed small RNA are predicted to target protein coding genes versus transposable elements?
It's a long sentence.
It is.
All right, yeah, okay, yeah, so good question. So what we did with the degradome sequencing is that you find all the ones that target protein coding genes, and that was that's what degradome sequencing do. It captures all the messenger cleavage bits. So in terms of the TE, I believe that degradome sequencing wouldn't give you that kind of data, but there are definitely bioinformatics software that you can do. I guess it would be considered as a computational method where you find the reference genome and use different platforms to identify the ones that could basically, silence the transposable elements. But that wouldn't be in our degradome sequencing data. You probably would have to design a different bioinformatics pipeline to do that, and then you'll be able to see the proportion. Yeah. All right. Hello?
Yep.
Yeah.
Yep. For geo analysis, did you use Waite term database or used other plant databases?
Oh, that's that includes other plants as well, because we wanted to just use wheat specific ones, but then that actually don't give you as many results because the wheat genome is just not as well annotated as other crop species. So yeah, is the other plants as well. Yeah.
And a question from Helena Oki. You have potential for 3-way interactions between treatment, genotype, and stage of development. You presented potential 2-way interactions, e.g. developmental stage and genotype. Did you do any formal testing of whether there were 3-way interactions?
The short answer is we haven't yet. So the 3-way interactions, that only works for the small RNA sequencing, because that's where we did the 240 libraries, where we included 3 factors. For the transcriptome sequencing and degradome sequencing, we actually only did day 5. Which is 5 days after thesis. Like, if I'm being honest here, is because of cost, because that's way too expensive for 240 libraries. If you want to do transcriptome sequencing, you probably need 5 Decra money to do that. So yeah, we definitely are interested to look at the 3-way interactions between the small RNA expression as well. And that's when I said we're gonna look further into individual models. That's where we're gonna get into that. It's just a shame that we wouldn't be able to do the same for the messenger RNA because we don't have 3 factors, data.
Okay, and a question from Melissa Garcia. How do you plan to look for association between the identified microRNA and phenotype in other durum?
That's a good question. So I actually talked about with Jason yesterday, right here on this spot. So what we have currently is that we have to remember we had those 10 genotypes that we did profiled in Glasshouse. And I actually have samples for all the 10 genotypes. It's taking pretty much the majority of -80 at the moment. So what we want to do is after we find the key ones, key microarrays I want to look at, we'll be able to go back to those samples. And those are the samples where we do have those 3 factors. So genotype, stress type, and time point. And I also do have all the data in terms of, chlorophyll content, stomata conductance, yield performance. So all of the data is there. So we'll be able to look at the on the molecular level, the gene expression, and try to associate that between different traits at different levels. And we have enough replicates there as well to hopefully make that association.
Thank you very much, Haipei, for speaking today, and
That's
thank
all right.
you everyone for attending.
Yeah.
Next week, we have Professor Tim Cavanaro speaking about Two Tales of a City, Carbon Neutral Adelaide and Urban Agriculture in Adelaide. So thank you, everyone.
All right. Thank you.